This paper presents the first volume visualization system that scales to petascale volumes imaged as a continuous stream of high-resolution electron microscopy images. This first sequence shows a scanning EM dataset of roughly 1 terabyte in size. On the upper part of the screen, you see the current resolution level of the data. Note that we do not need to pre-compute a 3D multi-resolution hierarchy like an octree beforehand. During raycasting, we detect missing blocks and construct them on the fly from the 2D microscope tiles in the required resolution. New blocks are then paged into a single large block cache texture on the GPU. On the top left, you see the size of this cache texture along with its current usage, which is largely dependent on the used transfer function. The next sequence shows a typical user session with our system. We have one main view and two smaller inset volume views for additional context information. The inset views use the same view direction, but a different zoom level and a different transfer function respectively. Additionally, it shows the current viewport of the main view as a green rectangle. EM data are very anisotropic, dense and noisy, and enhancing large structures like axons requires fine-tuning of the transfer function. Therefore, we simultaneously show a second, more opaque volume view, which greatly enhances the user's orientation within the volume. A novelty of our system design is that it is completely visualization driven. We restrict most computations to the visible volume data. Missing data are detected during volume raycasting as cache misses and propagated backwards for on-demand out-of-core processing. The next sequence starts with an overview of the terabyte-sized dataset. Notice the visible rotation of the stack due to the alignment of the original 2D microscope slices, as they have been registered to form a single 3D volume. Our infrastructure for fetching 3D blocks is not only necessary for direct volume rendering, but also allows for fast, oblique cutting planes through the volume. Another main feature of our architecture is that it decouples sample access time during raycasting from the size of the multi-resolution hierarchy. As opposed to traditional tree traversal approaches, we use a virtual memory scheme based on a multi-level page table hierarchy directly on the GPU. In this sequence, we demonstrate that our system is designed to accommodate for a constant stream of image data from the microscope, and that we allow for live updates of image alignment information. Here we see a simulation of constantly changing image alignments by jittering individual microscope tiles. Note that the colored EM tiles are quite large. Therefore, we divide them into smaller subtiles for better memory management and disk access efficiency. The last sequence demonstrates empty space skipping on an industrial CT dataset of a rotation sensor. Our system focuses on efficient rendering of EM data, which usually are very dense and do not exhibit a lot of empty space. However, we also support empty space skipping by marking individual page table entries as empty and skipping entire volume blocks during raycasting. For this dataset, this led to a 5-fold speedup in rendering time.